This is learning objective number 12 with Serena Singh, and in this video I will be explaining how Rflp analysis and PCR can be applied to the Human Genome Project. So first we're going to talk a bit about what the Human Genome Project actually is. So the Human Genome Project was an international scientific research project with the goal of determining the sequence of nucleotide base pairs that make up human DNA, and of identifying and mapping all of the human genome. It was completed in April of 2003, and the HGP gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. So here you can just see the banner used for the Human Genome Project, and a quote by Mr. Francis S. Collins saying, "What more powerful form of study of mankind could there be than to read our own instruction book?" So moving into our learning objective, what we first see in the learning objective is something called Rflp. Rflp stands for Restriction Fragment Length Polymorphisms. Now this type of analysis is very important for DNA fingerprinting. In fact, it's an old method used for DNA fingerprinting, and you'll find out why later on in the video. So here we have an acrostic. This will help you better understand the process and hopefully remember it too. So the R stands for restriction enzymes are used to cut DNA into F fragments, which are different L lengths, and exhibit P polymorphisms. So polymorphisms are variations in the DNA sequence, and in this case variations in length of the restriction fragments. Now for each person the length is going to be different because everybody has a unique set of DNA. So to kind of bring together what I said in my last slide, during Rflp analysis we see that restriction enzymes cut DNA into fragments. The fragments are then separated according to size by electrophoresis, which creates a unique banding pattern for each individual. To help show this, I have a little bit of a chart here which kind of shows banding strands between people. So say we have some sort of crime that has taken place, we have a blood stain that was, say, found at the scene of the crime, and we have our several suspects lined up here. So we have Bob, Sue, John, and Lisa. Now we use electrophoresis to create a banding pattern for the blood stain, and we do the same for each of our suspects. So now all we have to do is match up the banding pattern of the blood stain to one of our suspects, and we'll find out who it is, because as we said before, each individual has a very unique banding pattern. So I'll give you a moment to kind of look at it and see if you can figure out which one of our suspects is the perpetrator. Well, if you guessed John, then you are correct. If you look here, we see the exact same banding pattern for the blood stain and for John going straight across. So a little bit about electrophoresis. Actually, longer fragments of DNA tend to stay at the top of the band, while the shorter fragments come down to the bottom. So now we're going to talk about what we find in the second part of our learning objective, and that is PCR. So PCR stands for polymerase chain reaction. Basically what PCR does is it replicates a specific target segment of DNA many times. So while DNA cloning in cells remains the best method for preparing large quantities of a gene or DNA sequence, in the case of having scant or impure DNA, PCR is actually quicker or more selective. However, PCR does have its drawbacks, as it can sometimes make incorrect replications of DNA sequences, which is why cloning remains superior. So, with PCR, any specific target segment within one or many DNA molecules can be quickly amplified, that is, copied many times in a test tube. Now, the analogy that we have in Campbell's is actually pretty helpful. So what they say in Campbell's is that PCR is like photocopying one page of a book, or, one page out of, a few different books, rather than checking out the entire book in the library. So now that we understand the function of PCR, we're going to take a look at the process. So it takes place in a 3-step cycle, the first cycle being denaturation. In this step of the cycle, DNA is heated to separate the DNA strands. During the next step of the cycle, annealing, strands are cooled to allow primers to form bonds with ends of target sequences. During the final step of the cycle, DNA polymerase adds nucleotides to the 3' end of each primer, creating a full set of DNA target sequence. Now we're going to take a look at our diagram to my right. So if you look at this diagram of the polymerase chain reaction, you see at the very beginning we have our DNA, and on that DNA is the target sequence that we seek to make multiple copies of. So going down, we see our first step, which is denaturation. So these strands are heated so that they will pull apart and be able to separate. Now this is essential for the next step of the process, annealing, where cooling actually takes place. So the strands are cooled so that primers can form hydrogen bonds with ends of the target sequences seen here and here. In the third step of the process, extension, DNA polymerase adds nucleotides to the 3' end of each primer. So this is going to create our full set of the target sequence. Now if you go down, you see another cycle goes, we have these 2 replicated into 4, and in the final we have these 4 replicated into 8, and so on and so forth. And now to tie together everything we've learned in this video. So the aim of the Human Genome Project was to basically map out the human genome, or as some would call it, the blueprint of the human. Now the study of restriction fragment length polymorphisms is very important to the aim of the project because it helps us to study polymorphisms, otherwise known as variations within the genome. PCR is very important in working toward the aim of the project as well, because it's able to amplify or increase the amount of DNA. Now when we have more DNA available, this means that we can better study specific sequences of the human genome. By amplifying DNA, PCR allows us to more easily study DNA sequences of interest. And by studying restriction fragment length polymorphisms, we can better track the polymorphisms of the human genome.